Alright, assalamu alaikum everybody. How are you all? I hope that you all are doing well. Welcome to the class. Today we are going to be looking at AS level biology chapter enzymes topical MCQs. Right, today we are going to be solving the topical MCQs of the chapter enzyme. Right, so they are a total of 28 MCQs, I believe, which I have compiled from many years. It is from 2020 to 2012. Right, so we are going to be looking at all of these questions one by one, and then we are going to be solving these. Right, so let's now move on with question number 1. Question number 1 over here is from October November 2020 paper 1-2, which is question 14 in that paper. The question says that the painkiller ibuprofen reduces the activity of an enzyme involved in the production of the prostaglandin. Ibuprofen binds reversibly to the active site of the enzyme. Which type of enzyme inhibition describes this example and why? Alright, so we know that it is a reversible inhibition, right? We know that it is a reversible inhibition from the question. Then we know that there are two types of inhibition. One is a non-competitive inhibition and the other is the competitive inhibition, right? In the non-competitive inhibition, the inhibitor binds to the allosteric side. Inhibitor binds to the allosteric side, which is the side other than the active side of the enzyme, as well as the inhibitor is not similar to the shape. Inhibitor is not similar in shape to the substrate as well, right? These are the key points you have to remember for the non-competitive inhibition, right? And for the competitive inhibition, the inhibitor binds to the active side and it is similar in shape to the substrate and similar in shape to substrate, right? Now they are saying that which type of the enzyme inhibition describes this example and why. And in this example, what is happening that the ibuprofen binds reversibly to the active side of the enzyme, right? So ibuprofen is basically acting as an inhibitor over here, right? It is acting as an inhibitor because it is reducing the activity of an enzyme, right? So it is acting as an inhibitor over here and it is reducing the activity by binding to the active site. So it must be the competitive inhibition, right? It is the competitive inhibition. Now option C and D are incorrect. Now let's see that which option, A or B, is correct. Competitive inhibition, because ibuprofen alters the shape of the enzyme. No, ibuprofen will not alter the shape of the enzyme, right? It will not alter the shape of the enzyme. It will go and bind to the active side of the enzyme and will not allow the substrate to bind to the enzyme, right? And then in the B they are saying that competitive inhibition, because ibuprofen and the substrate cannot bind at the same time. Yes, it is correct. Why? Because the inhibitor ibuprofen will bind to the active side of the enzyme and will not allow the substrate to bind to the enzyme, right? So hence the correct answer over here would be option B, competitive inhibition, because ibuprofen and the substrate cannot bind at the same time. Now let's have a look at question number 2 over here. Now question 2 over here is from October November 2020 paper 1-2 question 12. It over here says that which statement describes some enzyme actions. Now they have given us the three statements and they are saying that which statement describes the enzyme actions, right? We have to choose the correct statements from these three statements, which are basically describing the action of the enzyme. Before we read all of these statements, we must know a very important fact about enzymes, that enzymes are known as the biological catalyst, right? They are known as the biological catalyst. They provide the sufficient energy for the metabolic reactions to occur. They speed up the metabolic reactions in our body. Without the enzymes, the metabolic reactions in our body would be too slow to sustain the life, right? So they are acting as a biological catalyst. We have to remember this statement. Now let's read statement 1 over here. It says that enzyme holds the reacting molecules so that their reactive groups are close together. Yes, this statement is absolutely correct. Why? Because we know that enzymes are the biological catalyst. They speed up the chemical reactions in our body. They speed up the metabolic reactions in our body. So it is very important for the enzymes to hold the reacting molecules or so that their reactive groups are close together so that they may react very quickly, right? And the metabolic reaction may speed up. So this statement is correct. Now let's read statement 2 over here. It says that in an enzyme catalyzed reaction, more molecules have sufficient energy to react than in the absence of the enzyme. Yes, right. Why? Because we know that the catalyst decreases the activation energy. And if it decreases the activation energy, which means that more molecules have the energy to react than in the absence of the enzyme, right? More molecules will be having the sufficient energy to react. So hence statement 2 is also correct. Now let's read statement 3. Reactions catalyzed by the enzyme take place at a lower temperature than they would without the enzyme. Yes, obviously, because enzyme is speeding up the rate of reactions, right? Enzymes are speeding up the rate of the metabolic reactions in our body, and they are doing it by lowering the temperature of the reaction than they would without the enzyme, right? Then they carry the reaction at a lower temperature, then the same reaction would be taking place at a higher temperature without the enzyme. I hope that you are getting what I am trying to explain over here. And yes, this statement is also correct. So hence the correct answer over here would be option A, 1, 2 and 3. All of the three statements are correct over here. Now let's move on to question 3. It says that what is the most appropriate set of controls to use in an investigation into the rate of an enzyme catalyzed reaction over a range of temperature from 25 to 65 degrees Celsius. Alright, so now over here they are talking about the control of the enzyme catalyzed reaction. So you must a control in the enzyme catalyzed reaction should account for the factors that might affect the reaction rate independent of the enzyme activity, right? So it can be done by using the substrate only at the all temperatures tested, right? Option D. How? Let's justify it. Like if you are using the substrate if you are using the substrate without the enzymes at all the temperatures, it will ensure that any changes observed in the reaction rate are due to the enzymes activity and not due to the spontaneous substrate breakdown, right? And yes, if the substrate itself break down at the higher temperature without the enzymes, it would give a false positive reaction rate in the experiment. So by testing the substrate alone at different temperatures, you can ensure that any product formation in the actual experiment is genuinely due to the enzyme action and not due to the breakdown of the substrate at high temperature, right? So hence the correct answer for this MCQ is option D over here. Alright, so now let's have a look at question 4 over here. It is Alright, so now let's have a look at question 4 over here. It is from February March 2020 paper 1 to question 14. It says that an investigation is carried out with an enzyme at its optimum temperature and pH. Optimum temperature and pH means that the pH or the temperature at which the enzyme is at its maximum activity, right? The rate of the enzyme's reaction is measured at different substrate concentration. You guys can see over here that different substrate concentration is seen, and the rate of reaction is measured at the different substrate concentration. By the way, this graph is not from the question. I have just added this graph from the internet so that I can better interpret you the question, right? The investigation is repeated in the presence of the competitive inhibitor. Competitive inhibitor, we know that a competitive inhibitor is an inhibitor which comes and binds with the active site of the enzyme and prevents the substrate from binding to the active site of the enzyme, right? How will the result with the competitive inhibitor be different from the original result? Now they are saying that what will happen with the Km with the competitive inhibitor and what will happen with the Vmax with the competitive inhibitor. If I talk about the Vmax, so you know that what is the Vmax? Vmax is the maximum rate of reaction, right? Vmax is the maximum rate of reaction. And what is the Km? Basically Km is basically the substrate concentration at half Vmax, right? This is the Vmax, this is the half Vmax. So the substrate concentration at half Vmax is basically the Km over here. Now if I talk about the competitive inhibition, so what happens in the competitive inhibition, that Vmax stays the same, right? The Vmax stays the same, and what happens is that the Km increases, right? And similarly for the non-competitive inhibition, what happens? Non-competitive inhibition, what happens is that the Vmax decreases, right? The Vmax decreases, this Vmax will decrease, right? The Vmax decreases in the non-competitive inhibition, and the Km stays the same. Km stays the same in the non-competitive inhibition, right? So what can be the correct answer over here? That in the competitive inhibition, the Km will increase, it will become higher, and the Vmax will stay the same. The rate of reaction, the maximum rate of reaction will stay the same, but however the Km will decrease, right? So hence the correct answer over here would be option B. The Km will become higher and the Vmax will stay the same. I hope that this is clear to all of you. Now let's move on to question 5. Now question 5 is from October-November paper 1 question 13. It says that the Vmax and Km are determined for an enzyme catalyzed reaction. Alright, so they are also asking us about the effect of the Vmax and Km in the presence of the competitive inhibitor, just like the similar question above. So Vmax, we know that the Vmax in the presence of the competitive inhibitor stays the same and the Km increases, right? So hence the correct answer over here would be option D. Like for example, you can see this graph, right? In this graph, this is the competitive inhibition, right? This is the graph when the competitive inhibitor was there, and this is the graph when non-competitive inhibitor was there, and you guys can see that in the competitive inhibition the Vmax is not disturbed, it remains the same, right? I hope that this makes sense. Now let's move on to question 6. Now question 6 over here is from October-November 2019 paper 1 to question 15. It says that when investigating the rate of reaction of an enzyme lipase on the hydrolysis of the triglycerides, which are the lipids, the pH must be maintained at an optimum to prevent the lipase denaturing, right? Now over here lipase is an enzyme which is basically hydrolyzing the triglycerides. Now they are saying what is the reason for this, that the pH must be obtained. We know that when the lipase is going to hydrolyze the triglycerides, so what is going to be the breakdown products which will form is the fatty acids, right? Fatty acids, 3 fatty acids and 1 glycerol molecule will be released from the 1 molecule of the triglyceride, right? And lipase is basically hydrolyzing the triglycerides over here, and lipase works at a alkaline pH. It is the property of the lipase that lipase works at an alkaline pH, and you know that as the lipase is going to hydrolyze the triglycerides, fatty acids and glycerol is going to be produced, and fatty acid will lower the pH, right? It will make the pH more acidic. So what can happen, that the lipase can get denatured. Why? Because the lipase is adapted, or the lipase has a characteristic to work in the alkaline pH, and fatty acids will lower the pH and make it more acidic. So yes, so hence the correct answer over here would be that what is the reason for to maintain the optimum pH for to prevent the denaturing of the lipase. So the correct answer for this would be that the products of the hydrolysis decreases the pH, right? Why? What are the products of hydrolysis? Fatty acids and glycerol, and fatty acids decreases the pH, it makes the pH more acidic and the lipase can denature in it. So hence the correct answer over here would be option B. The products of hydrolysis decrease the pH. Let's now move on to question 7 over here. It is from October-November 19 paper 1-3 question 11. It says that some extracellular protease enzymes, which are the protein digesting enzymes, are synthesized in an inactive form called the zymogen. Zymogen is converted into the active proteases in the Golgi body. What is the purpose of producing zymogen before converting it into the extracellular proteases? Alright, so basically they are talking about the zymogen, which is a protease in an inactive form, right? Protease in an inactive form, and they are saying that what is the reason that why the protease enzyme zymogen is initially produced in an inactive form before it is converted into the extracellular protease in the active form in the Golgi body, right? So basically why we are producing the zymogen, why we are producing a protease enzyme which is in an inactive form, it is to basically avoid the unwanted digestion of the protein inside the cell, right? Because we don't want that the zymogen may act on the cellular organelles and digest the cellular organelles, right? The protein parts of the cellular organelles. So hence the correct answer over here would be option A, to prevent, या फिर you can say that to avoid the unwanted digestion of the proteins inside the cells, to prevent the unwanted digestion of the proteins which may be useful to the cell, right? So hence the correct answer over here to question 7 is option A. Now in question number 8, which is from October November 19 paper 1 question 14, they are showing a graph. Basically the graph shows how the concentration of the components 1, 2, 3 and 4 of an enzyme catalyzed reaction changes with the time. Right now they are saying which row identifies the component of this reaction. You can over here see that the 1 is increasing, 2 is 3 is decreasing, then the 2 is decreasing and then increasing, and 4 is increasing and then decreasing. They are saying that what may be the correct representation of component 1, component 2, component 3 and component 4, right? They are giving us the enzyme substrate complexes, the products, the unbounded enzyme substrate, right? So now basically let's talk about first the mode of actions of the enzyme. In the mode of action of enzyme, there is first the enzyme and then there is the substrate. What happens is that first of all the enzyme substrate complex is formed, right? And then after the enzyme substrate complex, the enzyme product complex is formed, where the product is still in the active site of the enzyme and it is not released from the active site of the enzyme, right? And then what happens that the enzyme plus the product is there, right, when the product is basically released from the active site of the enzyme. So what will happen over time over here, that the product, the concentration of the product obviously will increase, right, when the enzyme catalyzed reaction will take place. Whenever the enzyme catalyzed reaction will take place, so what will happen over here, that the concentration of the product may increase, right? Concentration of the product may increase, right? And what will happen to the enzymes? If I talk about that, what is going to happen to the enzyme? So the unbounded enzyme, let's talk about the unbounded enzyme, because in the components they are using the word of the unbounded enzyme. First of all, as the reaction will start, so the unbounded enzymes will be decreased in number, right? Because there will be more substrate and more enzymes will be bounded to the substrate, right? And after the substrates are finished, so what will happen, that the unbounded enzymes will increase as the reaction is finishing, right? As all of the substrate are going to be used up and will be converted into the product, so what will happen, that the unbounded enzyme will increase. At the start of the reaction, the unbounded enzyme would be decreased in number, right? Because more substrate is available, so the enzyme may bound to that substrate. And at the end of the reaction, the unbounded enzyme may increase when the substrate is finished, right? And then if I talk about the substrate, so substrate will obviously decrease in number, right? Substrate will decrease in number. The substrate concentration will decrease as the substrate will be breaking down and be converting into the product. Now if I talk about the enzyme substrate complexes, so what is going to happen with the enzyme substrate complex, that the enzyme substrate complex will first of all increase, and then as the substrate will be about to finish, then the enzyme substrate complexes will also decrease, right? Because the enzyme substrate complexes also have to be converted into the enzyme product complexes as well. And also when the substrate will be finished, so obviously the enzyme substrate complexes will also decrease. Now let's interpret the graphs, right? Now let's interpret the graphs according to the information we are having over here, right? So if I talk about the product, so product is increasing, right? The product is increasing, so I think this one can be the product because the product is increasing, right? And if we talk about the substrate is, obviously we know that the substrate is converting into the product, so the substrate would be decreasing, so 3 is the substrate, obviously, right? And then if I talk about the unbounded enzymes, the unbounded enzyme will first decrease, right? Because there will be more substrate available, then as the substrate will be finished, so the unbounded enzyme will increase, so hence 2 is the unbounded enzyme, and obviously 4 will be the enzyme substrate complexes, which is first increasing and then decreasing. So hence the correct answer for question number 8 over here would be option D. I hope that this MCQ is clear to all of you. So question number 9 is the same question as we discussed above about the zymogen, right? So let's skip this question and let's move on to question 10. Let's now give question 10 a read, which is from February March 2018, paper 1 to question 13. It says that lysosomes contain many different hydrolytic enzymes that may act within the cell's intracellular enzymes or outside the cell extracellular enzymes. Now further in the question they say that which process must occur in order for the lysosomal enzymes to act outside the body. In order for the lysosomal enzymes to act outside the body, they must undergo the process of the exocytosis, right? In which what happens is that a vesicle comes and it fuses with the cell membrane, right? For example, over here if I draw, this vesicle comes and it fuses with the cell membrane of the cell, and then it releases its contents out of the cell. So hence the correct answer for question number 10 over here would be option C, exocytosis, right? Alright, so now let's talk about question number 11 over here. It is from October-November 2019, paper 1-3, question 12. It says that the Mitchell's-Menten constant for an enzyme catalyzed reaction is referred to as Km, and the maximum velocity of such a reaction is known as Vmax. Which statement about Km and Vmax is correct? Alright, so before we begin to solve this question, you must have a prior knowledge about Km and its affinity with the enzymes and the substrate. If the value of the Km is lower, it means that the affinity between the enzymes and the substrate is higher. Affinity between the enzymes and substrate is higher, right? And vice versa, that if the Km is lower, if the Km is higher, so the affinity between the enzymes and the substrate is less, right? So let's look at the options one by one and let's try to conclude the correct answer. An enzyme with the high value of Km can reach its Vmax at a low substrate concentration. No, it is not correct. Why? Because the affinity between the enzyme and substrate is less, which means that substrate is less effectively binding to the enzyme. So it may need a more concentration of the substrate to bind with the enzyme, right? And to reach its Vmax. So hence statement A, option A is incorrect. Let's now move on to question option B. It says that an enzyme with a high value of Km has a high affinity to its substrate. No, it has a low affinity to its substrate, as we discussed. An enzyme with the lower value of Km can reach its Vmax at a high substrate concentration. No, it is not true. Why? Because as the affinity between the enzyme and substrate is higher when the Km is lower, so basically what can happen that the enzyme with a low value of Km can reach its Vmax at the low substrate concentration as well. Then let's move on to option D, that says that an enzyme with the low value of Km has high affinity to its substrate. Yes, this statement is absolutely correct. So hence the correct answer over here would be option D, that an enzyme with the low value of Km has a high affinity to its substrate. Alright, so now let's move on to question number 12 over here. It is from February-March 2018, paper 1, question 14. It over here says that the diagram shows an enzyme, its substrate and an enzyme substrate complex. This is an enzyme over here. This is the substrate and this is the enzyme substrate complex. Then in the question it says that which statement explains how the substrate is able to enter the active site of the enzyme. We have two hypotheses over here which we discussed in our syllabus as well. The lock and key hypothesis, which states that the shape of the enzyme and the substrate are exactly complementary to each other, right? The enzyme acts as a lock and the substrate acts as a key. They say in the lock and key hypothesis, it is that the shape of the enzyme and substrate are exactly complementary to each other. But there comes another hypothesis which is the induced fit hypothesis. It states that the shape of the enzyme and the substrate, the shape of the enzyme's active site and the substrate are not exactly complementary to each other. Rather than that, enzyme slightly changes the shape of the active site so that the substrate may fit into the active site, right? And this is what we are seeing over here, that over here the active site is of different shape and over here it is of different shape and is allowing the substrate to fit into the active site, right? So let's read the statements one by one and try to find out the correct answer. Contact between the substrate and the enzyme causes a change in the enzyme shape. Yes, this is the correct answer, I believe. Why? Because you can see over here that as the contact is happening between the substrate and the enzyme's active site is changing the shape, right? So hence the correct answer over here would be option A. And the change in the active site of the enzyme is a slight change. It is not a major or a drastic change, right? So hence the correct answer over here would be option A. I hope that this MCQ is clear to all of you. Now let's move on to question 13. Alright, so now let's move on with question number 13 over here, which is from February March 2018 paper 1 to question 15. Over here they say that what could be used to calculate the rate of the enzyme catalyzed reaction. If we talk about the rate of the reaction, so it is the change in the concentration, right? It is the change in the concentration divided by time, right? So they are saying that what could be used to calculate the rate of the enzyme catalyzed reaction, the appearance of the product, the disappearance of the substrate, the Mitchell's-Menten constant Km. The appearance of the product and the disappearance of the substrate, which is basically acting as a reactant, can be used to calculate the rate of the enzyme catalyzed reaction. However, the Mitchell's-Menten constant, it cannot be used to calculate the rate of the enzyme catalyzed reaction because it is basically the substrate concentration at half Vmax. At half Vmax, so it has no link with calculating the rate of the enzyme catalyzed reaction, right? In enzyme catalyzed reaction, we can basically calculate the rate by putting the change of the product, right? Change in the concentration of the product over time, or we can also calculate the rate by change in the concentration of the substrate over time, right? So this is how we can calculate the rate of the enzyme catalyzed reaction. Hence the correct answer for this MCQ is option A, 1 and 2 only. I hope that this is clear to all of you. Now let's move on to question 14 over here. Question 14 over here is from May June 18 paper 1 question 12. It says that which word from the table correctly completes the paragraph about the enzymes. We know that enzymes are globular in nature, right? Globular in nature and they have a tertiary structure, right? When the pH of an environment is decreased below the optimum pH of an enzyme, the dash bonds between the adjacent dash groups holding the dash structure are disrupted. Alright, so let's look at the options one by one. Alright, so the bonds which are affected by the pH, bonds which are affected by the changes in pH are the two types of bond, which are the hydrogen bond and the ionic bond. Hydrogen and ionic bond, right? And these hydrogen and ionic bond are basically between the I group, right, which is the side chain basically of the amino acids. So basically hydrogen and ionic bond between the R groups holding the tertiary structure are disrupted, because I told you that enzymes hold a tertiary structure, right? So the correct answer for MCQ 14 is option A. Hydrogen and ionic bond are disrupted due to the decrease in the pH, right? And these hydrogen and ionic bond were between the adjacent R groups holding the tertiary structure of the enzyme, right? So hence the correct answer over here is option A. Alright, so now let's discuss question 15, which is from May June 2017, paper 1 to question 11. The following statements are true about enzymes, and then they have given us the list of the four statements, and they are saying that which statements are correct about the induced fit hypothesis. We have one, the lock and key hypothesis, and then we have the induced fit hypothesis. What do the induced fit hypothesis says? It has a statement that shape of the substrate and enzymes are not exactly complementary to each other, which means that the shape of the active site of the enzyme and the substrate are not exactly complementary to each other. Rather than the enzyme slightly changes the shape of the active site of the enzyme so that the substrate can perfectly fit into it, right? So this is basically the induced fit hypothesis, and then we are going to read the statements one by one, and then we are going to identify that which statements are correct for the induced fit hypothesis, right? So let's start discussing it. Statement 1 over here says that folding of an enzyme molecule causes the formation of the active site. This statement is true, but it is the general property of the enzyme. It is not specific to the induced fit hypothesis which they are asking in the question, right? This statement is true, but it is not true about the induced fit hypothesis. It is just discussing the general property of the enzyme. So we will cross this statement. Then we are let's read statement 2. It says that the shape of the active site changes to enable the substrate to bind. Yes, exactly, this happens. This is what we have discussed over here in these points as well, right? Temporary bonds hold the substrate in its active site. Yes, temporary bonds will be made which will be holding the substrate into the active site. This is an important part of the enzyme substrate interactions, right? So yes, this is also correct. Then the next statement says that more enzyme substrate complexes are formed at the optimum temperature. Yes, it is correct that more enzyme substrate complexes are formed at the optimum temperature. How they are at the optimum temperature? The kinetic energy between the enzyme and the substrate are increased. It leads to the more frequent collisions and more complexes are formed, right? So basically over here we can say that 2 and 3 are correct. But if we talk about 4, although it applies to the enzyme activity, it still supports the induced fit model because enzyme function depends on the successful binding of the substrate, right? So hence the correct answer over here would be option C, 2, 3 and 4 only. Alright, so that was it for the part 1 of this video where we solved the topical MCQs of the enzymes. Now inshallah in the next video we will be looking at more topical MCQs of the chapter enzyme and getting a more good grip on the other MCQs as well. Till then, have a nice day. Keep working hard for your upcoming examinations. And yes, if you guys did enjoyed solving these past papers with me, then do consider subscribing this channel and giving it a like. Thank you so much everyone.